Okay. All right. Well, why don't we get started here? Welcome back to the CFAR seminar series. And today we have two speakers. The first is Iain MacPherson, who will talk to us about exploring avidity in HIV design. And Iain will be presenting remotely from Zoom from Hawaii. So to just introduce Iain, Dr. Iain MacPherson is assistant professor in the Department of Tropical Medicine, Medical Microbiology, and Pharmacology at the University of Hawaii. He received his PhD in biochemistry and molecular biology from University of British Columbia, studying enzyme structure function using directed evolution and X-ray crystallography. And he postdoced at Brandeis University and Ludwig Maximilian University before moving to the University of Hawaii, where his current research focuses on on two main areas: aptamers, which are short pieces of RNA and DNA that have antibody-like binding function, and HIV vaccine design, which is the subject of his CFAR presentation. So without further ado, I'll hand it off to Iain.
Thanks for the introduction. As you heard, I went to UBC in Vancouver, so I've grown a great fondness for the Pacific Northwest. And actually, it was really rainy and dreary here this morning, so it almost felt like I was out there. I assume everyone can hear me. So I'll take it away here. So the top outline of my talk will be first go over the inspiration for the vaccine design that we worked on, and then I'll go into the vaccine design itself, then characterization of that design, and then hopefully there's time to talk about future work. Okay, so here's one of the big inspirations for this project is there's a paper that came out in 2019 out of the lab of Björn Hagborg. And Björn Hagborg's lab specializes in DNA nanotechnology. But what they did was something really interesting. They used DNA nanotechnology to space two antigens with real precision, and looking at different distances between those antigens and looking at the avidity of binding by a cognate antibody. So the antigens are nitroiodophenol, NIP, or nitrophenol, NP. And the antibody that they use can recognize both of those, but has a much higher affinity for NIP than for NP. And what they found was for NIP, the high affinity ligand, they saw that spacing was not as important for high avidity binding, for high avidity divalent binding. So that's displayed here. So anywhere between 5 and 16 nanometers allowed for high avidity binding. But when they switched to the low affinity antigen, the NP, they noticed that they observed that only with a defined spacing of 16 nanometers were they able to see high avidity divalent binding. And that the NP has a monovalent affinity of around 1 micromolar. So I thought that was really interesting, and I thought, hey, it'd be kind of interesting to make an antigen or make an immunogen that precisely placed two antigens, lock them in place in terms of orientation and position, and maybe that can serve as a vaccine. So this is really a big inspiration for that. So here I have the goal is to fix two antigens for optimal divalent binding by a specific B cell receptor. So the ideal scaffold that we're thinking of would minimize antigen positional and rotational freedom. So really, we're trying to lock two antigens in place here. It would also allow for antigenic spacing of around 14 to 16 nanometers. And we think that that will permit probably the highest avidity divalent binding, even for something with low monovalent affinity. And lastly, ideally, the scaffold would be minimally immunogenic. If you're making a vaccine, you wouldn't want off-target responses to the scaffold, ideally. And the effects of doing this by fixing these antigens is it minimizes the entropic penalty for divalent binding. because everything is sort of locked in place and ready to bind. There isn't much entropic penalty for that binding interaction. And this would promote high avidity binding. And also it would limit divalent, this is important, it would limit divalent binding to only a select few B cell receptors. So we think this might be a way to target specific B cell receptors and to convey a selective fitness for specific B cells. And this is the format that we've come up with. We've made a prototype, and it is a covalent immune complex. So here we have a complementary antibody that binds to a non-overlapping epitope on the antigen. And we've engineered a disulfide between the antigen and the scaffold antibody, the complementary antibody, to form a covalent immune complex. And the other design feature that's new about this, or novel in this, is we engineered a Fab-Fab disulfide. And the Fab-Fab disulfide effectively locks the Fabs in place for this divalent interaction. And because otherwise an antibody has, the Fabs have a lot of freedom of motion. And so we think that this Fab-Fab disulfide is critical in the design. So it's important to kind of frame this in the context of other immune focusing methods. So here we made a funnel, right? I've made a funnel, and the funnel, the top of the funnel represents all B cells that bind to a specific antigen, and different techniques or methods for immune focusing are shown on the right. So here we have hyperglycosylation, mosaic design, epitope masking, and heterologous boosting as methods that have been used for refining or whittling down that pool of B cells that binds to an antigen to that pool of B cells that binds to a specific epitope. And this is the next part here is where we think that our design may come in handy, is to select for the specific angle of approach to an epitope. And hopefully that can help drive selection for specific B cells to those that produce a target broadly neutralizing antibody. So here's, by doing this, with this design, we have to come up with some rationale as to why it might promote immune focusing. And the one that I mentioned already is entropic penalty minimization. So here I have the binding equation, and the more negative the delta G is, the higher energy that is released in that binding interaction. And as you go to greater entropy, so more disorder, that actually makes the delta G more negative. And what you want to do with binding interactions is minimize the amount of ordering that has to take place for a binding interaction. And by locking the antigens in place here, we've minimized the entropic penalty for the divalent interaction. Whereas if you have another B cell receptor that can bind to a different epitope, or bind to the same epitope, but at a different angle of approach that doesn't allow for divalent binding, that's going to require quite a big entropic penalty, because you're going to require two adjacent B cell receptors to diffuse into position to take part in that divalent interaction. We think that our design might have especially high immune focusing in the germinal center, so as a boost. And the reason for this is we think that it might convey higher signaling, B cell receptor signaling. So with this design, the pulling force you can think of as in the germinal center, B cells they exert pulling forces on the antigen to extract the antigen from the follicular dendritic cell. And in the case of our immunogen, the pulling force would be N. And in this case, for a non-BRCA, or I kind of gave away a little bit the future, but for a non-target B cell receptor, that pulling force is going to be halved, at least halved, because we already think that this interaction is going to be weaker than this interaction. So the pulling force is going to be a lot lower, and we think that that will enable a further immune focusing by conveying to the cell that this is a strong interaction worthy of further maturation of that specific B cell. So as I kind of gave away, we chose VRC01 as our model system, and VRC01 probably doesn't require much of an introduction to the UW CFAR because you have two of the best researchers in that field. But I thought I should just talk about it a little bit. So yeah, that's the model system that we chose. VRC01 is probably the best characterized HIV broadly neutralizing antibody. There are humanized animal models available for testing immune focusing approaches. VRC01 has been shown to confer resistance to an HIV infection. As you probably know, it targets the CD4 binding site of GP120. And the germline B cell receptor for VRC01 doesn't bind to native GP120. So this gave rise to the germline targeting approach, and which I'm sure many are familiar with. And specifically the N276 glycan is a roadblock that it keeps us from being able to completely generate VRC01 by vaccination. So I'll go more into detail about the design of the immune complex. It's based on antibody 48D. So 48D was isolated from a patient and structurally characterized, and it binds to the co-receptor binding site of GP120, less than 1 nanometer from the footprint of VRC01. And so here I have VRC01 modeled with 48D, simultaneously bound to GP120. And what we did was we engineered a disulfide between 48D and GP120, and that was using the program called disulfide by design, and it predicts cysteine mutations that you can make to form this disulfide. So these are the mutations here. And the other thing, when we modeled this, we were trying to figure out a way to fix the Fabs because we want to fix the antigen. And we looked at it closely, and it looked like mutating a specific lysine residue on the light chain of 48D might enable us to cross-link the Fabs. So here in orange, we have that loop, light chain loop containing lysine 126 mutated to a cysteine. So I have the cysteine here. But we thought, oh, this is kind of far apart. Maybe we need to extend that loop a little bit. So what we did was we modeled 3 other loops instead of that loop containing lysine 126 to hopefully create a better geometry for divalent binding. So we expressed these we purified using protein A chromatography and ran it on SDS page gel. And as expected, we saw a range of bands here. And this really boils down to, I'm going to run over this pretty quickly, but we have 0 bound to, the antibody 48D bound to 0 GP120s, 1 GP120, and 2 copies of GP120. So we wanted to figure out a way to characterize this immune complex. And I also work on aptamers, as you heard. So I developed some covalent aptamers that can bind specifically covalently to the FC of an antibody. And the nice thing about the aptamer is it allows us to both fluorophore label it easily, and also it is a huge negative charge, so it'll pull the immune complex through a polyacrylamide gel, a native gel, and allow us to track by a gel shift assay, track binding to a cognate antibody. So here we have a non-Fab cross-linked 48D immune complex and our model of VRC01 that bound to it. So here's the gel shift. So if there's 1 GP120, and this is incubation with wild type VRC01, when it's either 1 or 2 GP120s, we see a single shift here for the gel shift assay. And likewise for the Fab cross-linked version, K126C, we also see a single shift. So this is quite gratifying because it kind of suggests that we are getting what we're predicting. We're observing what we're predicting here. So we used as a control, we used an antibody called A32, which has a distinct epitope. It's not a VRC01 type of antibody, binds to a different epitope on GP120. Oh, by the way, I should mention we're using GP120 core in our design, but A32 binds to a different epitope on GP120. So we would expect if the fabs are not cross-linked, so they're waving around and they can rotate and also move laterally, that they would accommodate divalent binding by A32. And that's what we see. We see a single shift for either single or double GP120 immune complex. However, when we cross-link the fabs, make that K126 mutation, we would expect 2 copies of A32 to bind. And that is what we see. We see an extra shifted band here. I will add that we never see 100% cross-linking. So this actually has turned into sort of our benchmark for measuring cross-linking. And we've done this assay with the other loop mutants, and we can see that they're about 50% cross-linked. And the K126C, a single substitution, is closer to 90% Fab cross-link. So another interesting thing that can be done is try to incubate our immune complex, covalent immune complex, with a lower affinity version of VRC01. And our hypothesis would be that with a lower monovalent affinity VRC01, one revertants that we're working with that are closer to the germline VRC01. So sort of, reversion back in time in terms of the evolution of the antibody, that we might see a difference in binding between the Fab cross-linked and non-Fab. I have this, I apologize, I have this reversed. But in the Fab cross-linked version of our immune complex, we see a shift. And sorry, the labeling here is wrong. But for the Fab cross-linked version of our immune complex, we see a defined shift, whereas we do not see this shift for the non-Fab cross-linked. And this goes to the entropic penalty difference. So if we're locking the Fabs in position and the GP120s are locked in position, then they're better able to bind to this lower affinity version of VRC01, which we're calling 7-mute VRC01, and it's been measured to have a monovalent affinity of 152 nanomolar. So when we run the 7-mute VRC01 gel shift assay for the other loop mutants, and as well as their cysteine to alanine mutants that abolish Fab cross-linking, we see a higher binding for those loop mutants compared to their non-Fab cross-link versions. And it just, by looking at this, it looks like K126C, which we have in black here, and loop 1, which actually has the longest loop. They both seem to be doing better than the rest. So another thing we did for this immune complex is we fused. We thought, oh, it's very cumbersome to try to optimize the amount of plasmid that we're transfecting when we're, adding the GP120 plasmid plus the 48D plasmid to generate this covalent immune complex. We thought it might be interesting to generate a fusion. So we circularly permutated GP120, allowing for the C-terminus of the GP120 to be near the N-terminus of 48D. And then that's just one fusion heavy chain that we express. And this is what we get. So it simplifies expression nicely. And also, it also introduces some interesting things that we can do in terms of potentially trying to incorporate different homologous GP120s on each arm. We can talk about that later, maybe. Yeah, that's it. So we took this fusion construct, and we also ran another gel shift assay on that, using this time a 2 mutated version of VRC01. And this has a monovalent affinity of around 320 nanomolar. And so, but we also compared it with a low affinity version of A32, which we measured to be about 200 nanomolar. And the gel shift is strong, as you might expect, for the VRC01, with and without the crosslink, without and with the crosslink. And then for the Without the cross-link, this is interesting, without the cross-link with the lower affinity version of A32, we see a strong shift because the Fabs are able to rotate in our immune complex. They're able to rotate and accommodate divalent binding by this lower affinity version of A32. And for the cross-linked, Fab cross-linked version of our immune complex, we do not see this. So we're calling this this observation selective avidity because we are able to have a high affinity binding for a specific antibody having lower monovalent affinity than for a different antibody having a similar monovalent affinity. And these gel shifts are illustrated in these graphs here. So with the crosslink, you get almost zero gel shift, almost zero shift for the low affinity version of A32. So in conclusion, we created a covalent immune complex that fixes the positions of 2 copies of GP120 core for ideal divalent binding by VRC01. Fab cross-linking lowers the entropic penalty for divalent binding. And it also, Fab cross-linking also prevents divalent binding by non-VRC01 class antibodies. The low monovalent affinity versions of A32 bind much more weakly to the Fab cross-linked immune complex than to VRC01 revertants with similar monovalent affinity. So we kind of saw that as like a head-to-head. If you have 2 B cells whose B cells receptors have low affinity, can we get a selective avidity for the B cell receptor that we're targeting? And we think that this immunogen might have potential as an immune focusing vaccine. And if it does have immune focusing activity, then this design might be broadly applicable to other types of vaccines. So for future directions, what we'd really like to do is test our immunogens in vivo in a mouse model. And there are really nice mouse models available. And we would like to test, like the ultimate question that we want to answer is, does fixing the antigens for 1-to-1 divalent binding enable immune focusing? And we'd like to test it as a prime or a boost. And I mentioned that we think it might do better as a boost because of the dynamics in the germinal center and how there's more affinity discrimination by B cells in the germinal center. And we think that we might be able to make not only a monomer of the immune complex, but a dimer and trimer and still maintain selectivity that way. So we'd like to test those as well in a mouse. And our controls would be non-Fab cross-linked versions of these, with valency matched non-Fab cross-linked versions. And also we'd like to try a multimerized version of GP120 as as another control to compare and see, does our low valency antigen compete well against something that's multimerized. So hopefully the timing is good. I'd like to say thanks to everybody for tuning in or for joining and listening to my talk. Thanks very much to the Hawaii Center for AIDS, especially, well, everyone at the Hawaii Center for AIDS, but especially Cecilia Shikuma, the director. Ryan Bailey and Callista Kohoe Kapu did virtually all of the bench work that you saw in my presentation. And also thanks to the Department of Tropical Medicine, which I'm part of, especially Axel Lehrer, who's my collaborator, Ludwig Meyerlin and Albert Toe, who lended their expertise to this project, and all these for funding.
Thank you, Dr. MacPherson. So we have a little bit of time for a few questions. If anybody on the Zoom has a question, if they can raise their electronic hand, let me know, but otherwise we'll start here.
Hi, it's Larry Corey. I'm very intrigued by this because I think immune focusing is our
she got her PhD at Yale. studying regulation of virus-specific CD8 T cell differentiation in mice. Did postdoctoral studies at UCSF before getting appointed to the faculty. And currently the Rutishauser laboratory uses single cell profiling and functional genomics to understand transcriptional and epigenetic regulation of HIV-specific CD8 T cell exhaustion. And in HIV cure studies, the laboratory also applies systems immunology approaches to identify pathways correlated with immunologic protection in people with after infection and vaccination, with and identifying immune in identifying immune responses that may promote control of HIV after discontinuation of antiretroviral therapy. So without further ado, Dr. Rutishauser.
Thanks, Jay, for that introduction. And it's so nice to see so many of you. I feel like I've seen many people here and there over the last few months, and it's nice to be up here. So thank you so much for the invitation. I'm sure the weather's always like this in Seattle, right? Yeah, always, all the time. Okay. Just for me, oh, that's so nice. Yeah, I really brought it from San Francisco because so I started with what I have as a community slide, but I think also, I know this is a little bit more, some folks may be more general audience, but I think also helpful to just say in plain language sort of what I'm going to talk about today. So our lab, as Jay mentioned, we really focus on understanding CD8 T cell based immunity. And what I'm going to talk about is trying to answer the questions of how do HIV specific CD8 T cells in particular participate in an immune response against rebounding virus after antiretroviral therapy is stopped. Some of our adventures in trying to understand how therapeutic interventions might promote T cell proliferative capacity, which we think is an important capacity to assess in our studies, and then also try to elicit. And then sort of as a big take-home message, I'm going to talk about, we do a lot of like small studies to try to iteratively understand biology in different settings, kind of experimental settings in people. But I'm going to hopefully share with you two studies that give the same message, which essentially is that if you have a CD8 T cell population that's capable of having a good proliferative response as rebounding virus is happening, that can relate to later ability to control. And then some evidence that we have that new T cell responses to therapeutic vaccination in HIV might have better proliferative capacity. So we're in the HIV cure space, and I think thinking about how this relates in general to that research, I think a lot of literature, a lot of studies have promoted this idea that proliferative capacity of HIV-specific CD8 T cells is important, and I think we're contributing to that and providing further evidence that our therapies really need to focus on that capacity. So I, as Jay mentioned, I did my PhD with Sue Keck, who was just elected into the National Academy. Yay, Sue. So I'm obsessed with CD8 T cells, and I'm going to try to convince you to be as well if you're not already, but because of the work that we did in Sue's lab. So, in acute infections, whether in mice or in humans, CD8 T cells have this great capacity to be able to control infection. Of course, naive cells see their antigen, they proliferate, they differentiate into effector cells. Those cells can kill. Virus-infected cells, that's the goal. The majority of those cells die, and a small portion of those cells go on to become long-lived memory cells that have the capacity to be, rapidly reactivated upon seeing antigen again. So I left graduate school thinking that everything was LCMV, acute infection, and that CD8 T cells could just save the world. And the other thing I left grad school with was an appreciation that we can do things to shape this response. So we can give things different cytokines or different transcription factors can kind of shape making more effector-like cytotoxic cells, or we can shape the memory response in terms of promoting these memory precursor effector cells, these cells that have expression of transcription factors like TCF1 and FOXO1 that really allow them to be more likely to go on to become memory cells. But of course, reality is different. And in HIV, as in many chronic infections and in cancer, we know, this differentiation process is similar in some ways. Naive cells see their antigen, they turn into effector cells. Many of them, kind of contract after the initial phase. But when you have persisting antigen, of course, your cells become exhausted. This is regulated by some, transcriptional mechanisms, but is really an epigenetically controlled cell fate that's linked to kind of a distinct metabolic state. And then I think, as many people are aware, sort of expression of receptors on the cell surface of the CD8 T cell that promote or that inhibit signaling through the T cell receptor. And so this is one of the sort of challenges that we have in the context of HIV. But again, there's hope. So in chronic infections and also in cancer, we know that this whole population of CD8 T cells not every single cell is exhausted, but a subset of cells can express these markers, these transcription factors, and these regulators of proliferative capacity. And in fact, what we know from the last, I guess, 8 or so years now is that this subset, when you look at in mice or in people treated with cancer or with chronic infection treated with these checkpoint inhibitors, like anti-PD-1, that this subset of cells that expresses TCF1 that we call as sort of stem-like or memory-like, when they see that checkpoint blockade, they actually have proliferative capacity, and they have the capacity to sort of differentiate into secondary effector cells and clear cancer or control infections. So that's really what we're trying to understand in the context of HIV in our lab. And, what do we know about CD8 T cells? What do they do? And again, like, our work builds on the shoulders of many studies over many decades. But we think that CD8 T cells have the capacity to sort of make a dent on the virus, because we know that their activity is associated with, greater activity of the CD8 T cells is associated with lower set points in acute infection of where your viral load is, associated with lower set points after treatment interruption, but that they just might be coming perhaps too late to fully control. And then, of course, I think as many people here are familiar with, we have a model of individuals who naturally, spontaneously control infection in the absence of antiretroviral therapy. If it's undetectable, they've been known as elite controllers. Less than 1% of people infected with HIV kind of have this, people with HIV have this phenotype. And we know ex vivo, when you take their HIV-specific CD8 T cells, they're stimulated with antigen, they can robustly proliferate and they can robustly control virus in a dish. And that, is indicative of a response that exists in people who are naturally controlling. We see a lot of other aspects of their CD8 T cells and immune responses. But of course, the biggest, genetic association with elite control is also variants in class 1 alleles that, don't fully explain elite control, but could suggest the kind of potential effect on CD8 T cells. And then I think it's really interesting, I think over maybe more recent years, there have been more interventional studies that again have suggested that CD8 T cells might play a role in being able to control, whether it's the rhesus macaque CMV vectored vaccine, which generated HLA-E restricted CD8 T cells that were capable of sort of eventually clearing infection in over 50% of animals in a preventative setting, or in a couple of therapeutic settings that have suggested that CD8 T cells can control HIV. So I feel like we're at a place where we're trying to understand what can CD8 do, like how can we harness CD8 T cells, in rebound to control rebound. And there are, again, like to go back to what we've learned from elite controllers, elite controllers have these HIV-specific CD8 T cells that are highly functional. And to me, when I started my postdoc in 2015, I was like, these look like memory cells. These are really good memory cells, because when you stimulate them with antigen, they really proliferate and make secondary effector cells. And what had also been appreciated, and now we really understand is true as well, people who are not elite controllers, who had been viremic for long periods of time, of course, they have exhausted T cells. But even after you go on ART and you reduce viral loads, those HIV-specific CD8 T cells on a per cell basis are still functionally deficient. They still cannot proliferate as well. And so, again, building on all this work over time to understand, we've been interested in the cell intrinsic pathways that control this capacity in elite controllers. And so as a postdoc, I really focused on trying to understand the role of this important Wnt signaling transcription factor, TCF1. And we found, in fact, that TCF1 is more highly expressed in HIV-specific CD8 T cells from elite controllers. This is related to their expansion potential. It's correlated with a bunch of other memory markers like CD127, CCR7, et cetera. And that this high expression, in fact, goes down after you stimulate the cells. They become effector cells. So it needs to be a modulatable expression. And we further kind of dug deeper onto the transcriptional and epigenetic regulatory pathways that promote this functional capacity in elite controllers, together with Rafiq Sekali and Ben Youngblood, and really identified that HIV-specific CD8 T cells from elite controllers, they are, I wanted them to look like memory cells, like good memory cells, because they are so functional. I will say they are quite different from typical memory cells, but they do have this sort of TCF1 Wnt signaling stem-like program at the transcriptional and epigenetic level. And I was here last year giving a talk to the UW pathobiology students, so I'm now realizing this is a totally different audience. I presented on that last year, so I'm happy to talk more about it later, but I figured I would sort of summarize that work here, and then talk about some other questions in this talk. So we've been interested in these cell intrinsic mechanisms that promote, and everybody's probably seeing like, why does she have a cat there? But it makes total sense because HIV-specific CD8 T cells from elite controllers, they're ready to go. When they see a little bit of antigen, they go and they pounce, right? So they're ready to go, they're ready to pounce on that antigen. And so that's really the capacity that we're trying to harness. And so what I wanted to talk about today were some studies that we have ongoing in the lab to really understand, it's great, it's amazing to have the opportunity to study these cells that have this functional capacity and try to understand what makes them tick. But we really want to know in people, in vivo, can those cells actually participate in the control of virus as it's rebounding? And then we really want to think about sort of how do we apply this to therapeutics. So this is the first question, right? So again, sort of drawing the black line there of what the level of viremia would be in someone who was classified as an elite controller. The question is, if you put people who had previously controlled on antiretroviral therapy and took them off, can they recontrol? And a lot of some data in the literature suggests, people, when they come off ART, will just go right back to their set points that they have originally. But we didn't really know from prior literature that this would be the case with all elite controllers. We didn't know if, prior controllers would actually rebound always. And so we've had this incredible opportunity to study individuals in the kind of earliest moments of rebound due to this incredible study funded by the Gates Foundation and led by Lily Cohn, where, the purpose of the study was to try to identify, biomarkers of rebound. What's actually happening in the blood and then in the tissues and with the virus as rebound is occurring. So it's a very intensively monitored study in the blood and in tissues. But Lily and Steve Deeks and Mike Peluso at UCSF, as putting together the study, also designed it to layer in additional study participants who weren't just prior, what we say, kind of non-controllers, so people with high viral loads prior to ART, which is most people, but had been prior controllers, either elite controllers or people who were had low level viremia prior to ART, and then they went on antiretroviral therapy and they were taken off therapy. And the question here was, if you look at the earliest moments as rebound is happening, can you understand that to control HIV after treatment interruption, there would have to be a rapid, robust, and coordinated host response to rebounding virus? And we really think CD8 T cells are an important part of that, but we recognize, many other cell types are playing a part in that. So what do the actual rebound curves in this study look like, just to kind of give you a sense? So I think for folks working in the cure field, I think everyone's aware when ART is stopped, HIV viral loads, exponentially increase in the blood over the course of a couple days. I mean, they're very vertical lines, and this is a subset of the people who were sort of non-controllers in the study. Prior to starting their ART. When they came off, viral loads go well above 10,000 within, again, just a few days. So I'm just going to show some of the patterns from 6 of the controllers. So because there are a couple different patterns just to appreciate the heterogeneity. So 2 of the people who had been prior controllers, and I'm sorry for the color, I was trying to change it last night, but if you see in the pink lines, they actually went up pretty high and then came down and controlled. And that's actually sort of what can be seen in people who are classified as post-treatment controllers. There were people who went up to about a little over 1,000, a few thousands, and sort of kind of oscillated around 1,000. And then there were prior elite controllers who, had taken off bound. And you can see particularly one individual who very slowly went up and sort of very slowly came down sort of on their own. So it's a really fascinating study. No one has samples from individuals like this who have controlled virus, to some extent after treatment interruption at really close levels. And so we're so grateful for the opportunity to collaborate with Lily and folks in her lab who are looking at other aspects of this. The study I'm going to tell you about that we have been working on is that we decided as part of the study, there were lymph node fine needle aspirates, really trying to get a sense of what's happening in the tissues prior to treatment interruption, and then 1 to 2 weeks post-treatment interruption, right? So usually before rebound happens, but ART is worn off. And a lot of this was done with the kind of hope of finding biomarkers and looking at the reservoir. And what we decided to do when the study opened, I actually still remember these conversations, Lily, we were having on the phone. I was like, let's do 10x sequencing, right? Like just, this is a couple of years ago. It's like, let's try to get as many of these cells, sequenced as we can and see what we see. We didn't really know what we would see. So we have collected lymph node fine needle aspirates and done 10x sequencing on a total of 19 of these lymph nodes. But what I'm going to show you is where we have paired pre and post ATI samples that so far have been analyzed, because we've been sequencing kind of in batch. And all of this analysis was done by an amazing PhD graduate student in my lab, Demi Sandell. And so what we wanted to ask is sort of what are the immune responses that are starting to kind of percolate in the lymph nodes in this subset of individuals. So we have 3 prior non-controllers and 2 prior controllers. And so just to put a finer needle on, again, we're trying to understand the biology of what happens as, right before rebound or as rebound is happening. The post-ATI sampling window was, 1 to 2 or so weeks post-treatment interruption. And, we caught most people in the study here before they started to rebound, but here are 2 individuals who we caught as they were rebounding. One was a prior controller in red, caught at a viral load of, almost 6,000, and one was a prior that was the non-controller, and the prior controller was caught at a very low viral, at about 280 copies per mil. So what do we see in the lymph node? So I'm going to show you analysis of the CD8 T cells, which is what we've been focusing mostly on, and what we can see as the virus is emerging. So this is, kind of down selecting on just CD8 T cells and then doing a clustering analysis. For those of you who think about CD8 T cells, we can see clusters that kind of represent more naive or central memory type CD8 T cells, and we see clusters that represent more kind of effector-like CD8 T cells. But there's a really interesting cluster, so cluster 8, which has a subpopulation of cells with very high MKI67 expression, and this is a gene that encodes a cell cycle protein. And we can in fact see that cluster 8 really is the cluster that contains the cells that are cycling. And it's a very small cluster of cells. Remember, we're just catching individuals just as they're starting to rebound. This is just the 2 people who were sampled at the viremic time points. And you can see the cluster abundances here. There's a lot of interesting stuff that happens between the controllers and the non-controllers, but focusing on this one cluster, cluster 8, we observed that, whereas, the controller individual in blue on the right had a 17-fold increase in the abundance of this high, cycling population of CD8 T cells between baseline and day 16. They were at a viral load of 280, 17-fold increase in this cluster. The individual who was a non-controller, already going off to non-control, already was at, a viral load of almost 6,000, only had a 3.3-fold increase in this cycling subset, which is really interesting because I looked at those 2 individuals, and before we looked at the data, I said, wow, that person who's at a high viral load is just going to throw everything off because their T cells are going to look more activated. They're going to look more cycling. And in fact, what we're seeing is the opposite. So with these 2 individuals, we see that the individual who went on to control had a greater sort of increase in this proliferating subset of CD8 T cells. And this is reflected in this individual also by measures of greater clonal expansion. And I'm just showing that using a tool called SC Repertoire on the left. And then on the right, you can see the representation of, not surprisingly, individual clonotypes that are actually responsible for that look like they're actually expanding. And then if we look out across the 5 individuals, again, the very small study, but we see the same thing. So we have now 3 non-controllers in red, 2 of whom weren't sampled till, they were sampled before they started to rebound. They don't have an increase in their Ki67 expressing or MKI67 expressing CD8 T cells, but, it of course was before they rebound. But now we have the second controller who didn't rebound for many weeks later, but ART is worn off. So theoretically, you might imagine there's a handoff between the ART and the immune system. And now you can see an increase in Ki67 expressing CD8 T cells in the lymph node here as well. We've looked at this in a number of different ways, but this is one of the simpler ways to kind of see this phenomenon. And so small study, but like highly unique. We have more samples to look at. We have other cell types to look at, but we think that this is reflective of this cat-like quality, this pouncing quality capacity of the CD8 T cells to sort of jump on rebounding virus in this setting. And we're really excited to look at the additional samples and cell types and kind of put this together with the virology data and some of the blood data to really understand the, is this happening in antigen-specific CD8 T cells? Is this what's happening in terms of basically sort of at a clonotype level across the CD8 T cells? So that's small study, kind of lends some credence to the idea that, if you have CD8 T cells that we know in a dish were able to, respond very well to antigen, that, in vivo, those cells might, also be more highly responsive. And so, the question we might ask is, can we manipulate this therapeutically? And what does manipulating T cells therapeutically look like? Well, this group knows this more than anybody. I think, there's a lot of immunotherapy approaches being developed in cancer, where we can think about, overcoming exhaustion or boosting CD8 T cells or making new CD8 T cells. But essentially I put the magic wand there because like a lot of this is like, we don't really know the best ways to do this. And we especially don't, I think, know it in HIV yet. But what qualities would you want? You might want new specificities that can recognize HIV that's not escaped. You might want cells that are not exhausted, that have this proliferative capacity, and of course that are able to get to sites of reservoir persistence such that upon treatment interruption, they can pounce on the virus, they can proliferate, they can make secondary effector cells and hopefully kill those infected cells. So we've been very interested in understanding, kind of from a CD8 perspective again, can you do this with therapeutic vaccination? Can you change the quality of your CD8 T cell response with therapeutic vaccination? And so to do this, we have been really heavily leaning on samples from a study that we conducted in San Francisco and UCLA together with, again, Steve Deeks and Kara Chu, in which we used a DNA vaccine. It's a consensus immunogen. So it's a multiclade consensus immunogen developed by Dave Weiner at Inovio. It's a DNA vaccine with an IL-12 boost administered with electroporation. And the design that we chose to do for this study was essentially to ask whether, at the time, we designed the study in 2016, and at that time, I think we were trying to think about the idea that people might want to elicit T cell responses and antibody responses with the same vaccine design. And to do that, you would need things like targeting of GAG and POL, which are going to be more conserved potentially, and plus envelope. And the question here was if there had been evidence from preventative vaccine studies in HIV, but then also, mouse literature, that the more immunogen, the bigger your immunogen for T cells, the lower your responses are against certain parts of the insert. And so the question here was, if we included envelope sequences, would we detract from the ability to make T cell responses against the more conserved GAG and POL immunogens? So we had this study, we designed it first to ask that question, and then secondly, we layered in two cohorts of people with HIV on antiretroviral therapy. The bottom cohort there was a open label study, people with HIV who were treated early, so less than 6 months after HIV acquisition. And then the first cohort, cohort A, was actually people who were treated during chronic infection, so more likely the people we want to be able to give therapeutic interventions to. Randomized, so it's a placebo-controlled study, randomized to include the vaccine with GAG-POL versus GAG-POL-ONV. So we had this study. We had a good number of individuals in the study. We gave 4 doses of the vaccine. We had leukapheresis samples at baseline, and then 2 weeks following the last dose of the vaccine. And then the other great thing about the study, we didn't do a treatment interruption, but because we have the leukapheresis, we could also dig into questions of, really, are you eliciting new responses and what's the quality of those new responses? So the main outcome from the study, which is not yet published, but is something that we've reported at CROI, it's a DNA vaccine with electroporation. It did okay, eliciting responses, but we definitely saw that in the GAG-POL arm in red, So without the envelope insert, you could see a significant increase in the magnitude of the GAG-specific T cells, and this was true for Pol-specific T cells at the peak of the response, so week 14 on the left. And that you don't see this boost. I mean, you did in a couple individuals, but you don't really see it if you include the envelope immunogens, either in the chronic, so cohort A, or in the individuals who are treated early after HIV acquisition in green. And then same thing with durability, we saw kind of a trend towards maintaining these responses in GAG. We saw a significant increase at week 48 in the Pol responses. And, you know, these are a DNA vaccine. You have a lot of CD4 responses to the vaccine, but we also could see CD8 responses as well. This is another way of showing the same idea of kind of, again, the main outcome from the study, which was that if you were to add up who was a vaccine responder, which is actually very hard to do in a therapeutic vaccine study. So these are having twofold increase in overall to your antigens. You could see in the GAG-Pol group, about 70% of individuals were responders at week 14 in orange, whereas about 40% maintained that kind of twofold increase at the week 48, and that this was decreased in the GAG-Pol arms. I think this is an interesting signal from the study. It's their small study, but I think the people who got the vaccine who had been treated early after HIV acquisition, that is highlighted here. They're on the right side of the graph, but highlighted here, they actually, the same proportion of people remain responders at the memory time point. So it was not a very good vaccine, right? These individuals got the envelope included. It wasn't a very good vaccine. But to me, these data suggest that compared to the GAG-Pol ONV arm, so right next to that in purple, that perhaps people who get the therapeutic vaccine who didn't have a long period of untreated HIV, perhaps they actually are able to maintain memory responses better. And this hasn't really been explored in therapeutic vaccine studies, but it was part of our hypotheses in the study. So I think more to explore there. Okay, what about the antigen specific responses? So this is just showing cohort A individuals, placebo, people who got GAG-Pol on vaccine. And this is showing here that we did epitope mapping by ELISpot. This was done by Mansi Perwar at Wistar, where you have the individual participants along the bottom and you have the individual GAG peptides that we looked at along the kind of the y-axis. And I'm only showing here participants and or peptides that had responses. And what's really nice, and we're showing kind of the heat map is showing you the magnitude of the response at week 14. And what's in boxes are boosted responses, and what's in boxes with hashed are entirely new responses. And what you can see is the GAG-Pol arm had, you know, overall more participants had more boosted responses. And then what you can see with the hashtags is that, you know, more of the new responses also showed up in GAG-Pol arm. But what was really nice, when I call these new responses, I mean, they were not present at baseline. So we stimulated with the peptide for 10 days in vitro, came back, did another ELISpot. They were not there. So these are truly as good as we can get, we think, new responses. And, you know, how do we get at understanding what their functional capacity is? So I'll show you a little bit of work with tetramers, though it's hard to do that systematically across the study. But, you know, one of the interesting things about these new responses makes sense. They are present at lower frequencies at week 14. That's in the box in red. You can see by ELISpot that, you know, the new responses make sense, lower magnitude than the booster responses. The proliferation of those responses, you can see we did by a 6-day cell trace violet assay. So we're gating on the cell trace low populations, and we can see, you know, the stable placebo responses. It's a little bit of a messier assay, so not quite as stable, but in black. And you can see the boosted responses in purple. And what we see actually is that the new responses do seem to have a higher proliferation, higher proliferative response at week 14 compared to just the baseline stable vaccinated response. But what we really wanted to get at is, okay, these new responses are at lower magnitude, but they seem to have higher proliferation or similar proliferation. What we wanted to get a sense of is what the proliferative capacity of those responses are. And so we decided to do this. A couple others in the field have done this, to take a look at the ratio between proliferation, so the ratio of the, you know, percent cell trace low populations, thank you, over the magnitude. And here you can really see what you could intuitively tell from the previous graphs, which is that the new responses in orange have a higher proliferative capacity. You know, they are at a smaller magnitude, but if you were, you know, it's sort of like per magnitude, their ability to proliferate is probably better. And that's interesting because we think these responses are recruited from a naive population and therefore maybe don't have the epigenetic scars of exhaustion that we note in people, you know, who are on ART and experienced chronic infection. And so this was like, again, part of our hypothesis for this study. Hasn't been observed in other therapeutic vaccine studies, but we think points to the idea that, you know, there are a lot of complications about thinking about eliciting new responses. Will those responses actually see antigen that's presented in an infection? You know, all of that. But here, at least in terms of the quality of this response, this would suggest that these new responses might have better cat-like proliferative capacity. Sorry, I have a 4-year-old and she gave me a cough 2 months ago and it has not gone. I don't blame her. She's really cute, but yeah. So we are really interested in understanding sort of, you know, what is promoting that proliferative capacity and what are the mechanisms of, you know, even if you look at any of these responses, you can see there's a wide range for any antigen-specific response in terms of proliferative capacity. And so we're really interested in sort of understanding that more. So as part of the study and as part of a sort of sub-study funded by the Gates Foundation, we've had the opportunity to, you know, look at, you know, we have all this epitope mapping, we have all the HLA types, so we can look at class 1 multimers. And when we look at, you know, tetramer staining of HIV-specific CD8 T cells, one thing we can do is sort those cells. We have leukapheresis, we have lattice cells, we can do single-cell RNA TCR sequencing from these cells. And what's really nice when we do this, my lab knows that I really want them to get really good tetramer staining. Like we share this on our Slack channel, like how good is your tetramer stain? You know, so when you get really good sorting of nice tetramer populations across 2 time points in people who are placebo, you see total, this is looking at the TCR clones, you see, you know, total stability in those responses. It's a good gut check that, you know, if you didn't give an intervention, the clonotype distribution is similar. Most people have reported with sorted tetramer positive cells, HIV-specific CD8 T cells, that's this like oligoclonality. So most of our responses look like the ones on the left. The one on the right actually is a little bit more atypical for like a long-term response that's been sitting in the context of HIV for a long time. And, you know, some tetramer responses remained stable in magnitude despite the fact that we vaccinated them, which might mean that the responses didn't see antigen, or we wondered if that might mean they're more exhausted. So hold on to that for 1 second. The other thing that single-cell TCR, you know, RNA TCR sequencing allows us to do is identify some newly recruited clonotypes. So that may be entirely new responses like this one, which did not exist at baseline, wasn't there, but now, you know, clearly is there, and we can sort and see those clonotypes. Or within a response that you can see is very boosted by the vaccine, we can look at the clonotypes there and we can see, for example, the pink clonotype is particularly expanding, whereas the red clonotype, it looks like it's contracting, but if you look within the percent of live cells, it's actually just essentially sort of stable over time. So this gives us a chance to say, okay, we did the experiment. We have the experiment of we have some existing HIV-specific CD8 T cells. What happens when you give a therapeutic vaccine, and what are the T cells that are actually responding to that vaccine, which is analogous to, you know, what happens when the cells are encountering rebounding virus. So it's just a different kind of stimulation. But it's not stimulation in a dish anymore. It's stimulation now sort of in vivo. And this is just getting a window into some of the things that we're thinking about. But, you know, one of the things you might ask from this is what's different at baseline between the pink clonotype and the red clonotype, the really hyperproliferative one versus one that didn't respond. And we've done this kind of individually for a number of different antigen-specific responses and see similar profiles that the highly proliferative cells have higher expression of a lot of markers that we associate more with like central memory phenotypes. So I'm showing here SELL, which encodes CD62L, and LEF1, and then a number of interesting ribosomal proteins that kind of have been known to like poise memory cells for immune responses, secondary immune responses. And then what's up in the red clonotype, you see TIGIT and PDCD1, which encodes PD1, and CMC1 is a really interesting mitochondrial gene that comes up actually in exhausted cells, and its function has been defined to some extent in mice. So we're really interested in trying to think about what do these cell states look like of these cells that are, capable of responding to vaccination, and how can we think about, eliciting them or coaxing, or actually, Larry, I'm going to steal the word shepherding, if that's okay. It will mean something different here because we can't shepherd our TCRs, unfortunately, sadly, no germinal center, but we do want to shepherd, I think, a differentiation state into something that, might be more amenable to responding effectively to rebounding virus. So we're doing all this work kind of in the hopes that we can achieve that. So in the last little bit of my talk, I thought I would talk a little bit about a super interesting clinical study. So far, I've talked about CD8 T cells and how great they are, and trying to make CD8 T cells better with therapeutic vaccination. This is a combination immunotherapy clinical study that it's very funny. My graduate student, again, Demi, that's what happens when you have your first graduate student in the lab. She works on all the important things here. She was like, so we call it the kitchen sink study, but what does everybody else call it? And then she had a poster at Croy this year, and everyone was like, is this the kitchen sink study? And I was like, so Demi, this is the thing. When you work with Steve Deeks, he will tell everybody that it's called the kitchen sink study, and then that's just how it's going to be known. We're struggling with the paper right now. We're like, can't really call it that. But the idea here was that, from a lot of, again, building on the shoulders of many other studies here, that, individual immunotherapeutic interventions haven't really had an effect on rebounding virus. So what if we combine them? And NHP data, largely from Dan Baruch, had suggested that perhaps if you combined a TLR7 agonist and some BNAbs, you could get some effect on the reservoir. Perhaps if you combined a therapeutic vaccine with reservoir mechanisms, you could do something. And then, a lot of data indicating that giving BNAbs at the time of treatment interruption might, be something that would be interesting for inducing a so-called vaccinal effect. I didn't get into all that here because it's a lot of data to cover. And I think, at the end of the day, we had to do a combination therapy study that was, with GMP product that we could access, and think about sort of how to time this. So this was sort of the brainchild of Steve, and then was a study that was run by Michael Peluso and fortuitously opened in February of 2020. Great time to start enrolling your most complicated study, and included 10 individuals who are like heroes to us all for having kind of gone through this really intensive regimen. And I think are teaching us a lot about sort of what the regimen does. So these were 10 individuals, people with HIV on antiretroviral therapy. 7 had been given, started on ART relatively early after HIV acquisition, and 3 who had started ART late. And there were sort of 3 phases. So the first phase was a phase of, let's give a therapeutic vaccine. And the vaccine that we gave, thank you very much, Jim, was designed by Jim and has been studied by Barbara and Felber and George Pavlakis, as a conserved element DNA vaccine meant to really target very small regions within GAG, given intramuscularly with electroporation with an IL-12 adjuvant, and then a full-length GAG boost, and we had an MVA boost. So DNA prime, MVA boost, therapeutic vaccine, first stage, try to make some memory T cells. The second stage was this kind of trying to give a TLR, in this case, we had access to a TLR9 agonist, lefatolimod, and 2 BNAbs, 101074 and VRC07 523LS, with the idea being that perhaps this is still on ART, you would, maybe mediate some reservoir reduction, and maybe, although I didn't believe this, and I don't think we're seeing it, you would expand and maintain some effector T cells or NK cells kind of with the TLR9 agonist. You don't have any antigen around, so it's not clear what that is doing. And then this third phase where we gave the 2 BNAbs, ART is worn off, and then you get kind of the BNAbs wearing off over a long period of time. And then the idea with this phase was that perhaps under the cover of BNAb suppression, you could elicit, promote some kind of immune response as the BNAbs are wearing off that would eventually get ahead of the virus while you still have some control of the BNAbs. So, this study had 10 people. I will say, we recognize we didn't have a control arm. We have a lot of historical controls of people who were taken off of ART, who had been, started on ART early in their infections. So we know that we don't have a control, so we'll just keep that in mind. So, time to rebound, not surprisingly, was related to the pharmacokinetics of the BNAbs. When did the BNAbs wear off? And was the rebounding virus susceptible to the BNAbs or not? And so the people who rebounded early, I'm not showing the PK modeling, but this is just showing the time to rebound. People who rebounded earlier were rebounding earlier because the BNAbs were wearing off. The people who rebounded late were rebounding late because of still having BNAb on board, except for this one individual who we'll talk about who didn't rebound at all. So that was expected. What was not expected was that we think we're seeing very unusual kinetics of rebound in this study. So we have 10 people. We have 3 individuals who have what we call more or less like typical rebound, a more or less exponential increase in their HIV viral loads to very high, set points, and then they go back on ART. We had 1 individual who didn't rebound, and we think they have an unusual reservoir that will, we're in the middle of kind of characterizing. But what I think is really interesting is we have these 6 people who have kind of partial control. You have 4 people in blue who sort of their viral loads went up to 1,000 and then they sort of stopped. So looked a lot like 2 of the controllers in the ATI study. And they oscillate a little bit, and we kept, the study had long periods of time off ART, so we could really see where set points were and were they staying, so it's durable. And then these 2 individuals who took, many weeks to really even get up past 1,000, that dotted line, which you never see in the context of infection without any intervention. So we think we kind of have these 6 individuals who have this interesting sort of level of control. And I'll just show you, this is a very complicated slide, but for people who are kind of thinking about the PK of the BNAbs and thinking about what might be related to this is modeling done by Amelia Dykman. You can see in black and gray the PK models of what's happening to the 2 antibodies. We had one, again, that was longer acting than the other. And you can see sort of where the rebound is occurring, and across the top line, if you see red bars, that means that the rebounding virus had lost susceptibility to the BNAbs. And you can see, again, like I've annotated here that 2 of the people who rebounded quickly had started on antiretroviral therapy late. Despite that, the remaining individuals who had been started on ART early in their infection still have what we think is sort of a higher level of post-treatment partial control of HIV compared to kind of historical controls. Okay, so what did our interventions do? We don't know. I'm just going to preface this by saying we don't have all the answers right now, but I wanted to share some of these data because we think they're interesting. So the first question you might ask is, does the vaccine actually elicit a T-cell response? And we know from, this vaccine hasn't been tested in people as a DNA with an MVA boost. So it's, better than just DNA alone. And indeed, what we see is all individuals either mostly boost, or elicit new CD4, CE-specific CD4 T-cell responses. It's a pretty, CD4 T-cell response is a pretty profound boost. You have persistence at the memory time points, and you can see, you don't really boost your NEF responses on the right. That's our control to say, you're not seeing a boost in this vaccine phase of something that's not in the vaccine. The CD8 responses also are boosted for the most part. One individual did not have an increase in their or CE-specific CD8 T-cell responses, but at the memory time points in terms of magnitude, this actually wasn't above the baseline levels. So there are many phases to the study, and there's a lot of data I'm not going to, overwhelm your brains with. What we have found most interesting is to actually look at this question of what's happening as rebound is occurring and what are the immune responses that are taking part in that response. And so to do that, with this study, we have what we call the post-rebound 1 time point. It's the first PBMC time point that we have. It's within a month, oftentimes within 2 weeks of the rebound, but or 2 or 3 weeks within the rebound. All at relatively low viral loads, so mostly below 1,000 copy per mil, if not maybe 2 or 2,500 copies per mil. So very low viral loads, everybody at similar viral loads. And we wanted to ask this at a similar viral load for everybody, because once you get divergent viral loads and you look at immune responses, who knows what chicken or egg. So here, as the immune response is happening, before you get to your peak viral load, what's going on in the immune system. And so we've looked at a number of different parameters, but what I'm going to share here is kind of broad immunophenotyping of the PBMCs by CyTOF. And we're comparing this rebound time point to what's present at baseline and what was present right before you got rebound. And this was striking to us. So, we've looked across hundreds of different immune parameters. We looked across the ICS data. We've looked across a lot of different things. And really strikingly, we can see that at this early rebound time point, as the immune system is coming together, in the controllers in blue, you can see really, in some cases, very profound increase in the fraction of the CD8 T cells that's cycling, so that expresses Ki67. And that's happening in these controllers, despite, if you can appreciate here, slightly lower viral loads, actually. They're mounting this bigger proliferative response. And you can see that the non-controllers who already have, higher viral loads are already starting to go up here, have not increased the fraction of their CD8 T cells that's cycling. And so this is one measure of CD8 T cells that are responding to virus. It's in the blood. We see this in other activation markers. We see this in HLA-DR, CD38. We see this in other activation markers. And what's really interesting is these responding cells. We'll get to kind of what their phenotype is actually on the next slide. So we see this as a group amongst the controllers at this post-rebound 1 time point, and at this early rebound time point, we also see a relationship such that the individuals with the absolute lowest set point eventually wind up with the highest proliferative response at this early time point. So this again is like an in vivo setting now where we're seeing CD8 T cells proliferating in response to early reactivation or rebound that's occurring, and that preferentially the controllers are able to do that more than the non-controllers, although granted we have small numbers. The other really interesting thing is if you start to look at the phenotype of these cells in the controllers, so they've already started to mount this Ki67 response. I thought all those cells were going to be maybe more cytotoxic, terminally differentiated, but actually we see in the controllers, they have higher levels of expression of TCF1 compared to the non-controllers at that time point. And again, we see that relationship between the amount of TCF1 expression, so this transcription factor that promotes proliferative capacity, and low set point. We see a lot of other things in the CD8 T cell compartment at this time point. And one thing I just wanted to say, because I think a lot of people ask me is, well, what about the more differentiated cytotoxic cells? And in fact, in the controllers, we're also seeing a higher enrichment for those, but I didn't have time to I saw that yesterday and I didn't have time to put it in a slide for today. But I think there's, the CyTOF gives us a sense of what's going on broadly in the CD8 compartment, but we have a lot more work to do. So we have epitope mapping on these individuals. We're going to do tetramer work. We're going to do, TCR sequencing of the responding cells to really understand some of the questions earlier we were asking about the kind of trajectory of the clonotypes that are actually responding to rebound here. So from this study, I think, we have evidence of altered HIV rebound patterns with a therapeutic intervention, which has not been consistently seen with other therapeutic interventions. So we're really excited about that. Our early analyses are suggesting that, participants with better post-ART control have a more robust early proliferative response, and we have the additional analyses ongoing, not only of the CD8 T cells, but of course we recognize, host responses associated with control are going to be complex. So we have a lot of other assays, and there was a really nice discussion this morning at the roundtable about okay, well, how do we learn from these small studies, and how do we, we don't have a control arm. We do see some of these individuals. And I think, our approach here is this is a very unique study. We want to throw what we can at it to try to really understand the combination of these things and then sort of justify what the next study should be so that we can really understand, is this happening sort of across different studies and with different interventions. So I'll just leave with just, coming back to CD8 T cells and thinking about CD8 T cells. I have been very interested in obviously thinking about the basic biology of them, but then now also thinking about them in the context of vaccine studies and other therapeutic interventions. And I would say that I think what we've learned from our our in vivo studies of people with HIV coming off therapy and thinking about what responses are important. What we really need to be assessing is a functional response to antigen, functional proliferative capacity, ability to form secondary effectors, and our ability for, our therapeutic interventions to form these more memory-like populations that are eventually able to respond better. So that was a lot of stuff. I want to first and foremost thank all of our study participants who are just incredible to work with and get to know, and I think are really teaching us a lot about biology. All of our study teams, a lot of collaborators who I've mentioned throughout at UCSF, of course, Lily's lab here, and Jim. As well as Michael Gale as well here, and many other individuals on the individual studies. My lab is really wonderful, and I want to thank our funding sources. So happy to take questions.